Okay, let's do this. So this session is titled A Path Forward. And I think our goal for this time is to really leverage a lot of the discussions we've been having and start to target some short-term and long-term opportunities that we can start to take action on now. And we have a nice broad group of individuals here which bring many different perspectives. And so what I would like to do is maybe ask everyone just to start to introduce yourself and briefly in a minute, and then just mention your unique perspective that you'll be bringing to this discussion, where we're kind of trying to integrate everything that's been discussed and name some tangible next steps. So maybe we'll start with Robin and work our way this way.
Absolutely. Robin Patel from Mayo Clinic. I think that we're in a technology revolution, which is really exciting for those of us who work in diagnostics. But we're also in an AMR pandemic, and that's what we've said collectively today and yesterday at this meeting. Faced with the COVID-19 pandemic, we saw remarkable diagnostics development. We were told 276 molecular tests and 51 antigen tests received EUAs. I think we need to harvest that energy from COVID-19 and translate that now into this other pandemic of AMR. But I think we also have to recognize that AMR is actually a lot more challenging than COVID-19 in terms of diagnostics. We need to do this for our patients because we have to be able to get our patients on the right antibiotics they need and not antibiotics they don't need. And we need to invest in doing this in a very expeditious way. The challenge, though, for me is that diagnostics themselves are a means to an end, and they're not the end itself.
So my name is Jean Patel, and I am a clinical microbiologist. I've worked as an associate director of a clinical microbiology laboratory. I worked at the Centers for Disease Control and Antimicrobial Resistance, and now I'm a principal scientist at Beckman Coulter Microbiology, where I work on antimicrobial susceptibility testing. I think, again, thinking about the pandemic and how it has impacted our world, how do we respond today? A lesson learned I have is that increasing testing in the United States really required a public-private partnership in order to ramp up, and the faster that can happen, the better we can respond. I think, particularly in the U.S., we struggle with how to use diagnostics in an outbreak setting. And so you could think about an outbreak setting as a pandemic or a pandemic of antimicrobial resistance. But we were slow to pick up asymptomatic testing for antimicrobial resistance, and that's because we pay for diagnostics that will can be used to manage an individual rather than a population. I think that's another struggle that we have for antimicrobial resistance. I also think that science is leading us to strategies where we can identify infections before they can occur and use that information to prevent infections and to respond with the right antibiotics once an infection does occur. We need to figure out how to use that kind of knowledge to really be proactive in managing antimicrobial resistance for our patients.
I'm Susan Van Meter with the American Clinical Laboratory Association. I've also worked on behalf of diagnostics manufacturers in my prior role and spent many years working on behalf of New York hospitals and health systems after a brief time at CMS. I'd like to focus my comments just on thinking about, in a very practical way, how can we leverage existing infrastructure, policy infrastructure, to put in place additional incentives to encourage appropriate use of diagnostics, diagnostic stewardship, and to support a reimbursement environment that can help accelerate development of diagnostic tests. There's a lot to be said, but just a few quick things. We've talked about conditions of participation for Medicare that required antimicrobial stewardship programs in the inpatient setting. IDSA has done some work over time. I think they could do more. to bolster those requirements to really ensure that there are diagnostic stewardship of the variety we just heard about on the last panel. If we can build those diagnostic stewardship requirements into conditions of participation, I think that will help. I don't think COPs is actually the right place for this. They're often, they're updated maybe every decade. So we should think about ways in which we can have a measurement about having diagnostic stewardship programs in place that are truly meaningful as part of value-based payment initiative. That's a program that was launched by the Affordable Care Act. It basically holds back 2% of inpatient hospital payment. There's a series of measures, and you can be rewarded as a hospital both for advancement beyond your peers and for improvement according to your own baseline, right? When we think about healthcare equity issues we talked so much about today, we should make sure that we are not using incentive payments as really penalties, which is often the case. If there are hospitals in underserved communities that are struggling for resources, we want to make sure that we are meeting them where they are and providing the resources to be able to help improve the work that they do. We've seen that in the hospital readmissions program, where there are certain additional points of assistance for hospitals that had lower than anticipated, much higher than anticipated readmission rates. I think that is something that's doable, and we can take action on that. And also in the inpatient setting, we talked about NTAP, the new technology add-on payment pool, and the inpatient payment system. It hasn't been leveraged very much. T2 Biosystems was the only diagnostic company that was able to leverage it. We can actually increase that more. If we think about the outlier pool for inpatient hospital payment, that's really effectively leveraged by hospitals. We can increase that NTAP and put a focus on AMR. I think that would have some of the incentives, the net effect of some of the incentives we've been talking about. In the ambulatory settings, there is also incentive programs, the Medicare-based incentive program for physicians. We can have ambulatory diagnostic stewardship requirements in there that also reward clinicians for effectively putting in place these stewardship programs, particularly in the areas where we know there's the most problem with inappropriate prescribing, acute respiratory infection, and urinary tract infections. And then the Centers for Medicare and Medicaid Innovation, also part of the Affordable Care Act, has extraordinary authority to be able to waive rules and regulations to allow for pilot programs to experiment with different ways of delivering care. They are focused really on how can quality be increased while reducing costs, but they have further authority that actually allows them to increase quality and maybe even increase costs. Why don't we use that mechanism that exists right now, and it can be leveraged for both Medicare and Medicaid? And again, if we think about vulnerable populations and communities, right, leveraging both of those programs gets in an extraordinarily high proportion of Americans in communities throughout the country. I think a lot could be done with CMMI. There's the mechanism to do it, the existing authority. We should work together to push forward there some pilot programs that actually will help us develop the evidence that we know that we need to be able to communicate that value of using diagnostics for AMR across the patient population. I know we'll talk a little bit more about this, I'm sure, in Q&A, but we should really think at a very fundamental level, what are we doing in the U.S. to make sure we are continuing investment and laboratory infrastructure, not just for patient health, but public health in times of emergency as well. So there are some reimbursement challenges that are very significant, and we can work collectively to help improve those. Thank you.
All right. Thanks, everyone. My name is Rabi Shawar. I'm a branch chief in the Division of Microbiology and Devices at FDA in the Center for Devices and Radiological Health. Our division is the hub where everything came for COVID diagnostics, and I want to acknowledge and thank all the members of the division as well as all the members in our office and at FDA for their efforts on that. What we have learned from that is, and we knew that before, but the emerging technologies and how rapidly they are coming in to converge together, including diagnostic tools that require automation, that measure things digitally. They can look at things in a different way than we used to have. And so with that, for the pandemic, obviously the focus was more on rapidity, on getting the results and what have you. But this convergence of technologies brings its challenges as a regulator at FDA. One of the challenges is knowing what is the truth and finding what is the comparator reference method is going to be. And all of that is important to understand. But as we look at those, the developers themselves are the ones that know most about their technology. And when you bring that into FDA, for example, for evaluation, it would be best to outline all the details of that technology and help in order to get the studies in the right population at the right time and for right application. I want to give an example of something that came through as a challenge for us and for COVID, and then I'll translate that to some of the things for AMR. For example, new specimen types for diagnosing COVID, such as a breath. And when you thought about that, the only breath device that we had before, or I knew about, that's mostly used, is for urea breath test for H. pylori, which is a totally different paradigm. So I have to, put the thinking cap on and figure out how do we evaluate that, because it is actually measuring some markers and it is not necessarily measuring and analyze that we know about. What I want to end with is that because of these emerging technologies and many expertise are needed, collaborations become more and more important to bring in the parties that know most about the type of studies that may be needed, that have the facilities, that have the tools, that have maybe the specimen banks, that can come together in a collaborative manner. And I'll mention that later on maybe in one of the answers about how that could potentially be a recommendation from this group here. Because without the work collaboratively, we are really not going to be advancing the field as much. And on that, I will end with several of the collaborations that have taken place in the evaluations of COVID tests with places like the NIH, the NCI helped us in the evaluations. There are many collaborations that came out of that, and hopefully AMR can get into the game like COVID did. Thank you.
So good afternoon, everybody. I'm Joe Larson. I'm a VP of Clinical Development at Locus Biosciences. And it's exciting to hear about all these emerging diagnostic technologies that are all converging, and it's an exciting time for the diagnostic space. But there's also an equally exciting time in the antimicrobial space with kind of non-traditional therapies, and that's what we work on. We're basically applying precision medicine to infectious diseases through genetically engineered bacteriophage. So we basically engineer these phage, first of all, just to improve their lytic killing mechanism, but we also introduce different payloads, one of which being CRISPR-Cas3, which is, if you're not familiar with it, it's a component of the bacterial immune system that can recognize specific DNA sequences, target DNA sequences, and then basically acts like a buzzsaw and degrades that DNA. We have cocktails for staph, for Pseudomonas, for Klebsiella, thanks to CARB-X, and our lead program is in E. coli. We're currently enrolling a phase 2/3 clinical study in uncomplicated urinary tract infections for this E. coli cocktail. And immediately having a point-of-care diagnostic would rapidly help us be able to enroll that trial with a much greater efficiency than we currently are. In fact, we're using electronic health records to screen patients that have had previous urinary tract infections, and then the physician as well as the patient want to confirm that they do in fact have a coli before being randomized into the study. And so that adds a delay of 3 days before they can actually start being enrolled in the patient, and it's a 5-day long dosing in our trial. And so many patients have just said, no, I'll just take my antibiotics, thank you, and go home. And so that's been a challenge. And I think those challenges will further materialize with our Klebsiella program for UTI, just due to the proportion of Kleb versus E. coli that causes those UTIs. Another point I would make, improved diagnostics would clearly help us enroll trials better. But when you're dealing with our therapy has advantages in the sense that it's safer, probably less side effects than antibiotics. The FDA generally regards phage as safe. It also would minimize microbiome disruption and dysbiosis, which we think is a key advantage. But enabling all this and having clinical adoption of such a technology is going to require that diagnostics progress and then also equally become adopted as part of the standard of care. So over time, with some of these new therapies, you could imagine the paradigm of the linkages between diagnostics and these new therapies are going to be very, very strong. And I also will just bring up the other advantage we see is antibiotics, but in the context of my previous company I used to work for, which was Venatorx Pharmaceuticals. And we were developing a very broad spectrum antibiotic, cefepime, plus a novel beta-lactamase inhibitor called taniborbactam. And the advantage of taniborbactam is it was capable of inhibiting both serine and metallo beta-lactamases. And as we were completing our phase 3, we demonstrated superiority over meropenem in complicated UTI. One of the concerns about our commercial strategy was that, are we going to be withheld to only treat metallos? And I don't know if you're aware, but the commercial launches of new antibiotic agents over the last decade have basically been atrocious from a commercial standpoint. And so I'm going to take a second here and plug the Pasteur Act, which is up on the Hill, that would establish subscription-style contracts. We are very much in support of that effort. But in the absence of that, having diagnostics, and everything's been talked about here today, has all been about de-escalation. Well, in the context of a drug like cefepime taniborbactam, we would hope that it would be getting to the appropriate therapy as rapidly as possible, and that might help increase some of the poor commercial prospects and lack of investor sentiment that we're currently seeing in the antimicrobial space. So thanks to the organizers for having me. Appreciate it. Thanks.
Thank you, everyone. I think that's really helpful just to understand what your key insights are and what you're really bringing to the discussions. And so I'm going to challenge you even further. Susan, I think you did a nice job of laying out some specifics. And we've discussed a lot over the last 2 days. We discussed how do we quantify the value add that diagnostics bring, which is curbing AMR resistance, right? This is probably the biggest impact that rapid diagnostics can make over the long term, right? But I think we're starting to discuss some interesting ways of how we bring that back to an immediate, measurable metric. And so just on using that as an example and maybe diving in a little bit further there in reimbursement, I'd like to ask you all to kind of think of 1 to 2 really actionable next steps that you would prioritize of all the things we've discussed, right? Clinical guideline development, growing the energy in the global community through education. Maybe try and divide and conquer as you hear the ideas, but where should we start? And if you can categorize these, I'm going to try and combine the first two, into short-term and long-term actions, I think that would be, or goals, I think that would be helpful because I want to make time for the questions from the audience. Maybe we'll go backwards this time. Joe, do you want to go first? Or we can.
I mean, I am not a diagnostic expert, but from everything that I'm aware of or have heard, The problem of this is an economic one. I mean, it doesn't seem like it's a scientific problem at all. And so I don't know what policy would change, whose budget gets dinged for ordering a test, but it strikes me that some sort of reform, policy-based reform to the way these things are reimbursed would be an appropriate first step to increase adoption.
This is Ravi Shower. I cannot comment on anything that has any financial impact on anything, but I'm going to use this actually to answer something that I was going to mention it later, but maybe this would be appropriate, and that is the importance of collaboration. So at FDA, so this is from my perspective, at our center, there is something called collaborative communities. It is a concept that I think the cancer field, other fields have utilized that. The center put that out a few years ago, and it encourages, it is really an encouragement of various communities to come together, to put plans together, to discuss the problem together, much like this forum here. But when it's focused, say, on something, on an AMR, but specific 1, 2, 3 challenges that all the members of that collaborative community have to participate in and contribute to, that may be valuable. I'll send the link, or we can provide the place where you can nowadays you can just Google anything and find it. But that collaborative community, FDA is almost like an encourager, a cheerleader type of thing, where they participate in it and contribute in ways, but it's not driven by FDA. I want to make sure that it's not driven by CDRH. It is endorsed in some way. I hope I'm saying it right, but the website will clarify what that is. So I think that if any member in here is interested in that, initiating something like that, perhaps recommendations there can be given, and that may facilitate the process. Again, outside of what it might cost, I will leave that for others.
Okay, so first, in the short term, so if Congress doesn't intervene before January 1st, about 800 laboratory tests will be cut by up to 15% under the Medicare payment system. This would be the 4th year of reductions that have taken place. Within the last several. Congress has acted 3 times to hold back reductions. This is under the clinical laboratory fee schedule. We are hopeful that there is a bipartisan bicameral bill. You plug Pasteur, so I'm going to plug the Saving Access to Laboratory Services Act. It's salsa. It's easy to remember. It's a condiment. The CHIPS Act passed. I'm glad you mentioned that. Yes, it's chips and salsa. So we have a big campaign on it. This is borderline inappropriate, but stoplabcuts.org. You can take action. But the point of this is, right, for test developers, you need predictability. Medicare is a huge market. If there are continued reductions year after year on a relatively small baseline, right, we all know the statistics, right? Diagnostics account for 2 to 3% of total healthcare spending, but to drive somewhere in the neighborhood of 70% of clinical decision making, right? The investment in diagnostic is so worth it across the board, right? So alleviating those pending reductions, the SALSA Act doesn't even ask to remove them. It's about mitigating them to give a more predictable pathway forward through the clinical laboratory fee schedule that helps test developers understand. I mean, Joe, for your test in ambulatory settings, and or not for your test, for your drug, having the test that could go with it that would really help in your clinical trials and then be that proof point moving forward. You stand this risk with these reductions. So again, the SALSA Act is helpful. So that's one immediate thing that could be done and really needs to be done. We think about longer term, couple of things, right? I mentioned the CMMI, the Innovation Center, right? There is that authority, if we act collectively to encourage CMS to put in place both Medicare and Medicaid pilot programs that really leverage strong diagnostic stewardship across communities. There are Medicaid waivers that have community-wide performance metrics, these DSRIP waivers. Those are really important. Let's imbue some of those programs with diagnostic stewardship, right? These are things that are knowable and actionable and can be done with existing authority. And then finally, the ITAP program, I know I'm supposed to know what this stands for, but it's at NIH, it's the RADx program, but ITAP is, somebody can help me.
Independent testing?
That's different institute from.
Okay, well, we'll get it for the record. But ITAP has been enormously helpful during the pandemic. It was really established in partnership with the FDA to ensure that test developers could come through prepared to the FDA with clinical studies done on their rapid diagnostics. These are antigen tests, right? That model was great because it allowed test developers, both small companies, large companies, to be able to pull together their submissions to the FDA in a really expedited fashion, right? So if we use that mechanism, if we make it permanent and put funding behind it, when we know there are pathogens of concern, when we know we have this quiet pandemic, we can help ensure that NIH, in partnership with FDA, allows for a fast-track mechanism to get us the types of diagnostics we need as rapidly and appropriately as possible, whether they be truly point-of-care rapid diagnostics or laboratory diagnostics, which are, can help reach communities really anywhere across the country. So, thank you.
So we've been talking about rapid diagnostics for antimicrobial resistance, and we just learned a lot about the clinical impact of this, and this is improving antibiotic use. But who buys into improving antibiotic use and adopting a new diagnostic for that purpose? I think we have a new infrastructure in our country. Those are antibiotic stewardship programs. So now every healthcare facility is required to have an antibiotic stewardship program. But right now, there are no metrics for improving antibiotic use. And I think if we were to adopt metrics that hospitals would be responsible for improving, this would dramatically change how diagnostics are used in a laboratory. I think this institutionalizes the benefits of improving antibiotic use. There is not a case-by-case or a hospital-by-hospital case that has to be made, and it would be much easier to implement that kind of testing.
We didn't key this up together, but I'm going to completely agree with the other Dr. Patel. So, I mentioned that diagnostics are a means to the end, and the end is really the utilization of antibiotics here for these particular these particular diagnostics. And I think we have to recognize that we're not using antibiotics as well as we could in healthcare, and that we can make that situation better with better diagnostics. However, that benefit is to the individual, but it's also to the people around that individual and to society at large. And the way that we pay for healthcare and the way that we value healthcare is really around the individual. So it's important to recognize, I think, the societal benefits of doing the right thing with our antibiotics. We still need to use them. We're not going to get rid of antimicrobial resistance. This is going to be with us. Unfortunately, I wish we could come up with a strategy to end the pandemic. That would be wonderful, but that's not happening. But we really, this is, I will say, I'm an infectious disease doc, a medical doctor. This is a bit our fault that we got into some of the pickle we're in with antimicrobial resistance. And I just feel that with rapid diagnostics, we can do better, but we need, as Dr. Patel mentioned, to recognize that that's valuable, that using antibiotics correctly is valuable, and that there can be harm to the individual and to society by using antibiotics when they're not needed. And rapid diagnostics can really help us with that. I think we heard about some other strategies. We heard about guidelines, so I'm not going to go over that as well, but there's some opportunities to move forward there. I think investment in science is still needed. Antimicrobial resistance is not completely understood. There will be new mechanisms, and we need to stay on top of that. And we also heard from Dr. Banerjee about implementation science studies. The availability of rapid diagnostics in different settings is going to change fundamentally the way we deliver healthcare, but we need to figure out what that means and how to do it and how to get the most value out of that. And that means implementation science studies like the clinical outcome studies that she highlighted. And we need investment to support those studies because, as also pointed out, diagnostic manufacturers are typically not going to be able to pay for those, and that's important. A couple of other reflections. That one I don't think came out at this meeting. We did talk about the complexity of the term AMR. I think it's even more complex because as an infectious disease doctor and clinical microbiologist, we're talking about multiple bacterial species. When I teach our medical students microbiology, I say that there are over 500 bacterial species that are pathogenic to humans, and yet we're lumping them all here in addition to lumping all the drugs together and all the clinical syndromes together. I mean, a UTI and a respiratory tract infection are not the same, and also the sites of care that we heard about here. So that's a lot. I think we're going to need different solutions for each of those. We can learn, of course, they're not going to be 2,000 different solutions, but there will be many different solutions we need from diagnostics. And just as an example, the bacterium that we heard about today that Mallory was infected with, Burkholderia acinocepacia, it's not on CDC's list of antibiotic resistance threats. And there are many others that are missing there as well. So that could be an area to focus on. I don't want to leave something out just because it's not on, like the top 10 list somewhere. For the individual patients, antimicrobial resistance is a problem, even if they have somewhat of a more esoteric organism compared to the general population.
Amazing. Thank you. Just one thing I wanted to flag. Coming back to the really understanding how large this problem is in the U.S. and abroad, leveraging the data that we do have as these newer technologies become available and their real-time reporting resistance, and it's possible now. And how do we leverage some of the data banks that BD and bMaria and Beckman Coulter have to help expedite or accelerate some of these studies, right? I think we know where resistance is high. How do we use the data that we do have while we're trying to improve and leverage mechanisms in place to do this better in the future to be more efficient and perhaps expedite some of the clinical work that I still think needs to be done. So just putting that flag. I'd like to just use the rest of the time to open this up to the group here and also virtually. So are there questions in the audience? Okay, go ahead.
Nick Halzak, Roche Diagnostics. I had one comment and one question. So one comment, something that's been talked about a couple times is NTAP, and I just wanted to put a slightly finer point on that. My personal view, this is my personal view, not the view of Roche Diagnostics, is that the NTAP program, there's not nearly enough incentive at the other side of that to make that an effective program. I know that was kind of alluded to yesterday, but circumstances in which a provider would receive the pass-through payment are pretty rare. For diagnostics, at least, maybe not so much for devices, but for diagnostics, the actual dollar amount associated with it is relatively low. And even if there's enough there, it expires in 2 years when the fee schedule gets updated. So I think there is an opportunity to kind of bolster that program a little bit and try to remove some of those challenges. So that's the one comment. The other question that I wanted to raise, I think it's really for Susan, but others as well, it was very briefly raised on a panel yesterday, is this upcoming transitional coverage for emerging technologies pathway. It's something that I'm monitoring really, really closely. It would apply to diagnostics as well as devices. Susan, I know you have some history with this. I'm hoping that you could talk a little bit about that, what you're looking for in that program and whether it might be able to apply to AMR type products.
So the goal of the potential program is to help technologies, medical devices, which include diagnostics now, those that receive breakthrough status from the FDA and are cleared or approved by the FDA, to be able to have the opportunity to develop evidence while being covered, right? So coverage with evidence development in essence, right? So that can provide an enormous opportunity to medical device manufacturers. When we think about, and you would probably have the statistic off the top of your head, but there is a fairly large proportion of breakthrough technologies actually that are diagnostics. So they are in fact included in TSET. So that could create an opportunity for a certain subset of diagnostics to be able to be covered while securing evidence. It doesn't ensure that there's going to be a reimbursement rate that is going to be appropriate, right? It doesn't guarantee a market either. So I think it's an interesting idea. It's one that I think that commercial manufacturers are really interested in. A little bit of a different perspective from the laboratory view when we think about laboratory developed tests. But I think it's part of a larger conversation we should be having about evidence development. We need that evidence. How can we work in a collaborative way with government to be able to generate that evidence? I'm going to go back again and say, what about that CMMI, the innovation program? Can we use certain levers there to help develop evidence?
Yeah, since breakthrough was mentioned, I think it will worthwhile mentioning something for the audience, especially online. FDA has a program in my division, in our center, of course, as a whole, where we receive a pre-submission, and it's designated as this is a pre-submission for a breakthrough designation. Depending on the what is provided, an analysis takes place for different criteria, whether that does or does not meet, and once that's evaluated, it's either granted or denied. And the more elaborate information is there, the more there is evidence about the device in its final form, the more information about its technology, the better the designation would be, and it's all about benefit, risk, and what that device might bring. So just wanted to clarify that, what that is about. And in addition, there is a program, there is pre-submission program. Many of the industry here know about that, but for those who don't, the pre-submission program is a program that is now even enhanced more at FDA and with more focus on it as a program. We have MEDUFA 5 come through now. And it is a very efficient tool to bring to FDA with questions. And the more you describe about their technology, the better the answer would be. And it will help in guiding through, especially for newer technologies, where, like I said earlier, I mean, everything is merging and converging together. And so for that pre-submission, I hope that many people can take advantage of it, so that you can bring in innovative things and conduct the studies in the right way with the right reference method, et cetera, that we all can agree on. Thank you.
You know, I think this discussion of innovative diagnostics is important. And an example is Dr. Banerjee earlier mentioned the need for diagnostics that detect colonization, not just infection. And I think that is part of the science, that we understand that colonization precedes infection, and being able to detect a pathogen as it overpopulates a microbiome and is then a threat for causing an infection is a way that we can better conquer antimicrobial resistance. So you can, for example, you could detect overabundance of a pathogen in a GI microbiome, and that's most likely a cause of a healthcare associated infection. And so being able to predict that and either prevent it or be able to prescribe the right antibiotics, because you know what the pathogen is and you know what it's susceptible to, that is a powerful form of clinical care that might be very important in an era where there are few antibiotics to choose from. I think we heard a compelling case this morning of colonization preceding infection in a cystic fibrosis patient. And what if Diane's daughter had received phage therapy at the first site first indication of B. cepacia colonization that could have changed the course of her disease. And I do think this is an emerging area of medicine. I think, the program you just discussed where transitional technologies, I agree there needs to be a broad spectrum of those kind of programs so that we can develop diagnostics alongside therapeutics as clinical care practices are changing and evolving.
Thank you. Any other questions in the audience? Andrew, go for it.
Yes, so someone online is asking, how should we think about prioritizing the development and uptake of rapid diagnostics for the diversity of pathogens and care settings that exist?
I'll jump in. I'm probably the least qualified on the panel to jump in, but why should that? Let me get in. So if we think about sort of like the, and Jean and I were talking about this a short while ago, in ambulatory settings, the diagnoses where we have the highest levels in inappropriate prescribing of antibiotics are for upper respiratory infections and UTIs, urinary tract infections. So if I'm thinking about the ambulatory settings, I would prioritize those areas as being the ones where if we can better utilize existing diagnostics and help support the development of new diagnostics, we can have the biggest bang for our buck.
Yeah, I think simpler, faster tests are needed. I think we need to work hard to train folks on tests that currently exist. Not only how to use them, but how to use the results that come from them. I think one of our challenges that we face today in microbiology is that the microbiology lab staff has been decimated because of COVID-19. There is a need to replenish that staff. We can't permanently go on with a diminished technical expertise in infectious disease diagnostics. I think this is a responsibility that falls on all sectors of diagnostics and patient care.
Yeah, Paul.
Paul Etter, NIH. So after 2 days, I thought there was a certain word that wasn't going to be used, but Jean, you used it this morning, and that was asymptomatic. The potential value, maybe it was mentioned yesterday, the potential value of asymptomatic monitoring. And that might not sound cost effective in certain environments, but in, for example, retirement homes. And I noticed all this during COVID, not with the residents getting SARS infection, but rather getting UTIs that would lead to sepsis. And nobody could see any of that because they weren't being monitored. I mean, some of you are probably very good at monitoring what are the key things that where sepsis onset is coming. But nevertheless, so 2 areas of innovation might be asymptomatic testing and also wearables, where in places like nursing homes and things, they could be monitored. And then tended to much more quickly get the right antibiotic much more quickly. I'd like to see more investment in that. Actually, there is some investment in that. I'd like to see more in the government. Thanks.
Thank you, thank you, Jean, for bringing that. This brings me to one point that I wanted to also mention about when devices are being designed, to always think. We heard about the target product profile from the user side, and from, say, a regulatory side, we talk about the intended use. And so all of those really translate into what patients I'm going to go into, what are the specimens that I'm going to obtain, how I'm going to interpret that, and what is the potential action that may take place. So for screening, for example, for nasal colonization, skin colonization, whatever, there are tests like that, and of course it depends on the study you want to make, but also the claim that you want to make. And that is, if it's only for screening, just determining colonization, there obviously will be actions from that, and that could be just the interpretation of it. So I wanted to make that point. Another, while I have it on, that's it. I'm not going to speak one more time. I feel like I'm, someone like the cheerleader with pom pom. I'm going to make. I make about collaboration in general. Data, collaboration on available data. We live in an information age of I cannot describe the proportions of the information age that we are in, and that is scientific information that we're talking about. So how do you collectively utilize that? And to give an example of that, sequence data, genetic information, its correlation to phenotype, the level of evidence that you are obtaining. Is that 5 minutes for me to speak or for the whole session? Thank you. So that data piece information and gathering all of that, getting it for utility of for the public good, collaboration would be very important. And the other, which ARLG, the AMR Bank, other places, is the specimen and the sources. So that's not information, but it's sharing of other things with known information about them. And the more you know, the more you have characterized, the better when you utilize these studies, for example, to supplement prospectively conducted studies. And if you have specimen banks, if you have isolates, you can spike into, the field is open for how you could potentially evaluate the device perhaps in a more expedited manner, especially, for example, for rare pathogens and rare resistant organisms. And that becomes very important to, bring resources together on that. Thank you.
Thank you, Ruby. I do think it's helpful to think, we are kind of trying to boil the ocean here when we think about AMR, and I do think it is helpful to think about, what syndromic areas should we focus guideline development on? Is sepsis really meaningful because time matters so much, right, in terms of disease progression and patient outcomes as time goes on. So I do think it's helpful for us to I won't put anyone on the spot, but just think about how can we start? Where do we start in terms of a pathogen-specific approach, syndromic approach, or other focused approach? In the last 2 minutes, just any final parting thoughts? I feel like this is our closing session. We have a few wrap-up comments, but really, any other closing thoughts or inspiring words or major lessons learned that you just want to flag as we wrap up?
Well, one thing I've heard several speakers allude to is that this antimicrobial resistance is a global problem, and if we can't just improve diagnostics where we are today, but globally. And I think that's a huge challenge. We know that the health crisis is coming. It's just a matter of how bad is it going to be. And the sooner we can make sure everyone has access to diagnostics, I think the better off we'll be.
I wanted to thank the National Academies for putting this on. I think this has been an impressive 2 days and a great conversation, and I hope we can continue this conversation and work together. I think I was quite touched by the two ethicists, including Nicholas Evans, commenting that bioethicists don't really address AMR because there's not a lot of controversy. I do feel there is controversy based on what we've heard, so I hope bioethicists step up. And Tracy Cohen was very motivated by saying that some of the things that we are doing in clinical practice today, we medical doctors, prescribing antibiotics when they're not needed, prescribing the wrong antibiotics, delaying antibiotics, withholding them for fear of overprescribing, those are violating bioethical principles and duties of doctors and laboratorians, interestingly, as she also mentioned. And I think we need to consider this as we move forward. I think it's our ethical prerogative to act here.
Mm-hmm. Absolutely. So I think we have some buckets, right? We have how do we build demand? How do we educate the community? How do we use more congruous language in order to do so? And how do we tell stories to really represent what is AMR? How do we come together as a community to focus our discussions so that we can start to move the needle in meaningful ways without, again, trying to boil the ocean? I think we've talked about the economic incentives. At the end of the day, it's a money problem. So until we're either making money or saving money by using these tools in a meaningful way to really improve the appropriate use of antibiotics, it's going to continue to be an uphill battle. I think there's some behavior change that we've discussed that really needs to be a focus. And again, collaboration. I think, different groups are doing different things, and the more conversations we have and the more opportunities we can link together and work more efficiently, the faster this will all move, even though it feels like we're moving at a snail's pace. Kent, did you want to make any wrap-up statements? Okay, great. I'll hand it over to you. Please just stay. Yeah.
Well, who knew that a workshop with the very bland title of Accelerating the Development Uptake of Rapid Diagnostics to Address Antibiotic Resistance, a workshop, would have so much rich discussion. It's really, it's quite amazing. I really want to thank, of course, all our speakers, presenters, people that pose questions, comments, and perspectives. It's really important. And I think that as much as we think about, well, we're kind of moving slowly, it's only through discussions like this that we can try to mobilize things, people, organizations, and the public to make a difference. I really want to thank our various National Academy staff that have helped out behind the scenes and within the scenes today and in the lead up. And of course, all our members of our planning committee and my co-chair, Betsy. So I'm not going to recapitulate everything, but a few key themes that came out in this whole concept of rapid diagnosis. And that is the value to patients and to patient care, both in terms of providing timely diagnosis, leading to timely and appropriate treatment. The impact on the larger area of AMR, whether we then fine-tune it to antibiotic stewardship, diagnostic stewardship, and more, and also leveraging the data that's just bubbling out all the time. Terabytes of data are coming out in all different sources, and how do we get our arms around that? Some of the technical aspects. Do we want the quick wins? Do we want to just focus on sort of a binary readout? There's presence of an AMR determinant in a culture or in a colony, or do we want fine-tuned diagnosis? And does that matter? And if it does, in what case? Issues related to development and implementation, accessibility of the technology, the concept that diagnosis is part of our critical infrastructure, however we determine that. And I think and I think probably many people here have been in a variety of conferences, meetings, and other workshops related to AMR and antibiotic development, diagnosis. I think this is the first one where we've had extensive discussion on bioethics and health equity. And I think that's really important because that's just another aspect that has not been addressed in the past. The need for practical aspects, guidelines, the regulatory implications, policy. And policy is money, policy is utilization, and more. And then, of course, undergirding all this is cost, as Betsy just said. Cost of development, cost of deployment, if cost of the developer, cost of the public. So these are really important. And then I think finally, that was really highlighted earlier today, is the public awareness. We've been focusing on rapid diagnostics, but I think we see this as a continuum from just the basic, if you will, antimicrobial resistance to development of antibiotics to diagnosing those infections and following them through, hopefully, to appropriate treatment. So how do we stimulate public awareness? The stories, the advocacy, and ultimately action. I don't think any of us wants to walk away today and say, well, this was a really great meeting. Let's do it again sometime. I think there's much more important things at play. And I think, it's on all of us. And how best to address that, I guess we'll have to see. So again, thanks so much to all. It was a really good day and a half session. And thanks for coming.
Just a reminder that the recordings and the audio will be posted on the NAS website within the next couple of weeks. And also, there are copies of Salt in My Soul out in the foyer for everyone and a loved one or colleague. I think there are extras, so feel free to snag those on your way out.